I am delighted to be introducing Dr. Megan Herting, who will be our speaker today. And she is currently associate professor at the University of Southern California, as you can see on her slides, and the director of a neuroimaging lab. And with an important focus for us that is really very relevant to our own interests, which is environmental and lifestyle factors that influence brain and cognitive development in children and adolescents. She got her BS in biological psychology at the University of Illinois Urbana-Champaign, a PhD in behavioral neuroscience from Oregon Health and Science University, and her real claim to fame, and that she's going to be speaking to a lot today, is investigating air pollution effects on neurodevelopment. And she uses multimodal MRI methods, which is state of the art. She is co-PI for an important project at the Los Angeles site for adolescent brain cognitive development study consortium, co-chairs an environmental working group for this domain of work, and is co-founder of the Enigma Environmental Working Group to examine environmental impacts on brain structure across the lifespan. I think it sounds fascinating. So, Dr. Herting, I think we're grateful to you for having signed up to speak today. And at the same time, I want you to know that this is our Weathering Change seminar series, which has just started. It's only a few weeks old in terms of actually enacting it. So you're one of our early speakers. I think you have a climate group out there at, yes. And we don't yet know them very well, but we know them somewhat, and are going to be collaborating more over time, we hope. But just to know, this is one of the few Midwestern climate centers, research centers, or now we call them heat and extreme weather centers. So we appreciate the opportunity to have inputs from you. Thank you.
Thank you. Thank you for that warm introduction and the opportunity to present today, and congratulations on your. I'm looking forward to sharing with you some of the work we've been doing and tell you the story of how I got here and what we're finding in the data set. So thank you again. I always start off, I have no disclosures. My lab, as alluded to, is interested in what influences how our children's brains develop. And I'm interested in a number of exogenous and endogenous factors that influence these processes in the brain and risk for cognition and mental health outcomes in adolescence and into adulthood. And today I'll be largely focusing on some of the work we've been doing in terms of environmental neuroscience, studying how air pollution affects the developing brain. So I've been interested in the developing brain for as long as I can remember. And I'm specifically interested in child and adolescence because it's a unique period of vulnerability and opportunity. So we know from the animal literature and human studies that a lot of the brain gets programmed prenatally. And so a lot of focus has been on the DOHAD hypothesis, or the developmental origins of health and disease. But the brain is actually very malleable for a long time in terms of synaptic pruning, getting rid of and refining the processes and connections between the brain, myelinating or insulating these connections, and really spans a number of experience-dependent plasticity, or neuroplasticity, if you will. And so this now has come out a long time ago in 2018, but arguing that the new adolescence period is probably from ages 10 to the mid-20s, and it might now even extend beyond that in terms of brain plasticity. And so I've been very passionate about understanding what during this period of refinement in the brain might be influenced by various endogenous and exogenous factors. I also, from a kind of a sociological or psychology perspective, really also enjoy this period because it's a period of growth, physical maturation, hormonal changes, pubertal maturation, things that I've studied in a past lifetime, as well as lifestyle changes. These are some of the times in which kids stop physical activity or they decrease their physical activity. Nowadays, right, there's also a lot of changes in screen time, drug and alcohol initiation, and even just autonomy of self, and also their mobility in their environments. Socially, right, it becomes a time of peers and less about family and more about peers and self-identity. And then for the cognitive and the social emotional functioning, this is where planning and goal setting, decision making, executive functioning, and even emotional regulation, skill sets that really last us a lifetime in our day-to-day functioning. And so this is a really unique time for really pruning these patterns of not just behavior, but also at the brain level. In addition to all of these changes, it's thought to be a time of also vulnerability because there is a mismatch in the timing of maturation of different brain systems. Emotional and reward systems come online pretty early, and then the regulation through the prefrontal cortex of these behaviors is still in play throughout this adolescent and early adult period. And one of the statistics that always caught my attention was the fact that most mental illness begins, or at least early signs of it begin early during adolescence. So 50% of all lifetime mental illness tends to have some sort of onset by age 14, suggesting this is really some developmental origins. And so really starting to think about how can we understand individual differences and understand risk and resilience. You know, this has led me to the child and adolescent years as a focus of my research. In order to study these questions, we do a multimodal neuroimaging approach. Here's some pictures of some of the techniques we use. We can look at structural MRI for shapes and sizes of brain features. We can look at diffusion MRI, which is a water technique. It actually looks at water diffusion rates in the brain to get ideas about microstructure and connectivity between brain regions. And then we can also do functional MRI to look at changes in blood oxygen level dependence to get some information on possible brain function and network organization. And a lot of my work has tried to move beyond cross-sectional into longitudinal differences and really understanding individual variability over time. And so that's something that I've been passionate about regardless of the questions that we might be asking, but as more of an approach. Multimodal MRI has been really useful for understanding key changes that occur in the brain. This has been going on now for 20 plus years, knowing that the brain seems to peak and prune during the adolescent years when you study it with MRI, which is shown here. And we also know that white matter structures are starting to really refine themselves, and we can look at this with diffusion MRI, as I mentioned. We think that this might be in conjunction to some of the cortical thinning or pruning that happens during this time, as well as the white matter connectivity or myelination and increase of support cells. And so these typical patterns really worked itself out so that we can start to understand what various risk and resilience factors might be doing to these processes. Now, when I started this work, I thought, you know, the individual variability that we were seeing was because we were largely working with smaller cohorts at the time and a lot of cross-sectional data. And so a lot of my interest was also in longitudinal change. Of course, we found out that those patterns, which are shown here in an example by this Nature Neuroscience review, is that even though there are typical averages that we like to plot, that the individual variability is quite drastic. And so this really is, you know, an interesting thing that not just do we see cross-sectional effects that are widely variable, but we also see the trajectories and the change over time and brain maturation to be variable. And what does that mean for when people are at the time of increasing or stabilizing or decreasing their connections between brain regions, and how might make individuals might make certain individuals more vulnerable than others. So this body of work of studying child and adolescent brain development using multimodal techniques and really being passionate about some of the first, you know, studies to try to harmonize across different samples to try to look at longitudinal behavior with Kate Mills and others was really a landmark opportunity when I was a postdoc to participate in what is known as the Adolescent Brain Cognitive Development Study. So also alluded to, this is called the ABCD study. Many of you might have heard of this study before. It's the largest long-term study of brain development and child health in the US. And what we did is we recruited about 11,000, over 11,000, almost 12,000 children when they were ages 9 to 10. as a cohort, and from 21 different research sites across the country. And the baseline assessment happened during a 2-year window, in which that was between 2016 and 2018. And this includes coming in parent and youth for annual cognitive and behavioral assessments for the children. We do brain MRI every 2 years. The study visits can last as long as 8 hours. We collect information on almost everything you can imagine. And it's a longitudinal design. So we're almost at the end of our 10 years, and we're just about to put in our 5-year renewals to follow them into young adulthood. So those should be coming on the horizon as well. So as I mentioned, 21 study sites in the US really provide this wonderful diversity, both of the participants themselves, but the geographic coverage and the spatial differences in which we see people participating in the US. I am just one very small part of this study in that I oversee data collection at the CHLA USC site. But together we've harmonized this approach and follow the same protocol with everybody for the last 9 years now. And this started out as an NIDA project, but then of course the valuable data set and the scope of it really got a lot of exciting federal partners involved to support this research, which we thank. So as I was coming to terms with thinking about, individual variability, a number of factors, studying sex and puberty in individual trajectories, as well as having a history, which I'll talk about just briefly, in lifestyle factors, I was a little amazed when I read the general aims of the project in terms of experience and environment. So you can see here, one of them was that we will determine how childhood experiences and environmental factors interact with each other to affect changing biology, affect brain development, social, behavioral, academic, health, and other outcomes. But what you'll see here is the examples. So in cognitive neuroscience and behavioral neuroscience, in which my training was in, a lot of the environmental and experiences that most people were talking about included lifestyle factors, sports, video games, social media, and then, maybe going out one step further into socioeconomic status as an environmental factor. But, from my background, I had learned that there's really a lot of differences in how you study environment, which is probably something you also realize, given your center and the expertise there. But I started to think, what do you mean by environment? I had started my work in thinking about environment and neuroplasticity way long ago as an undergrad in Bill Greeno's lab. We were the people mapping how neurites changed to enriched, different enriched environments. So if you will, putting rats in different cages with different toys every day, and also social interactions and seeing how much more the brain could kind of adapt and proliferate. After that, of course, then I went to Oregon Health and Science University, in which my thesis work looked at how aerobic fitness and physical activity really influenced the adolescent brain, translating some of the work from animal models, but also aging into development. Of course, this was a great place to also, study physical activity. I was much more active back then, but running around in the beautiful environment of Portland, Oregon, with healthy green space and air. I couldn't help but also notice that when I started to do my work and started to think about environment, I was surrounded by wonderful colleagues now here at USC who were studying environmental health, if you will, right? A term that really my field wasn't really thinking enough about, stopping really at the lifestyle and maybe the social environment, but really ignoring a wealth and a body of literature that now is very well integrated into thinking about the built environment and even the natural environment. And of course, living in Los Angeles at that time, even just being aware of the surroundings, it was a different physical environment, and air quality is something that our university had excelled in showing was really problematic for lung function. And so all of this was kind of culminating together. The ABCD study had starting to think more about environment, talking with environmental health scientists and experts that I now are able to have the fortunate ability to collaborate with, and really started to get more and more interested in environmental toxicants, Again, speaking to the choir here, but we know now, right, a lot of different toxicants affect not only the child's body, but also the brain. They come from a lot of different sources, and they can influence the central nervous system in many ways to be potential risk factors for behavioral dysfunction and change biology. So with all of this exciting work going on, we wanted to start to think about how we could leverage the ABCD study to start to ask some of these questions. And so that led to the development of what is known as the Linked External Data, or the LED Environment and Policy Working Group for ABCD. I have had the pleasure of leading this group since probably 2019, and I lead it now with Dr. Cardenas Iñiguez, who's the co-chair and also now an assistant professor here at USC. Again, the wonderful work about this, right, is that we have the diversity in a population study like this in order to study variability that otherwise is hard to capture or disentangle, at least, when you just have one study site. And so really the geographical and spatial distribution adding to the value of saying, what might this mean for local effects or population effects, and how might there be heterogeneity in those patterns? And so we are now one of many working groups to do this work. And a lot of our efforts the first couple of years were revamping the geocoding and the address histories and in order to be able to say something about the environmental context in which kids live. Now, of course, this was an add-on later on, wasn't funded originally, and also was not something people were interested in adding to the actual battery themselves. So we get a lot of questions, you know, do you have actual monitoring of the air pollution or other environmental toxins on the individuals? Or do you know information about X, Y, and Z through questionnaire? And I wish we did. But one of the things that was able to sell this idea to the consortium was, other than collecting great addresses, we could use kind of more of a geospatial epi approach in order to add context without adding additional hours to an already 8-hour battery. So our goal as a working group is to enhance and try to provide a deeper investigation of some of the physical and policy also related variables that could influence some of the outcomes that we're studying in the ABCD study. We then subsequently got some grant funding thanks to NIEHS, which really has made this all possible, in which we were able to improve the geospatial accuracy, the temporal attributes, and really build really solid residential history for participants at the participant level. Dr. Cardenas Iñiguez then led us through really a rework of both the existing linkages that people had kind of initially linked to the data, but also new ones to really come up with what he calls the adolescent urbanome, in which he's linked and describes a number of various features to the environment and the built environment available in ABCD. But my passion and the reason that I wrote my 2 grants in the first place, coming back to this environmental question, has really been air pollution. So as many of you are probably aware, right, air pollution is a ubiquitous exposure with harmful health impacts. And so when I started the LED working group, I really wanted to study the question of air pollution. And as many of you are also probably aware, right, we can describe the mixture of gases and particulate matter that we breathe in many different ways. Our work is largely focused on fine particulate matter, given that EPA regulates this, and there are guidelines from the WHO. We have also focused on our own work in nitrogen dioxide and ozone as well. As some of the key ones, although there's a number of other gases that also are of interest. And really a lot of cardiometabolic, pulmonary, and in the last 10 years even, the central nervous system are really targets of this air pollution. I love this graphic from Mount Sinai because it really gives an overview of not just the types of exposures, but the various sources they can come from in air quality, as well as the various health effects and some of the risk factors. But we know that this stage of child development is in fact a vulnerable period. And we also know that exposure is, 99% of the world is still living in places where we're not meeting the WHO air quality guidelines or suggestions. And so really this is a ubiquitous problem and ever present. So our work has really then started to focus on PM 2.5. As I mentioned, we do look at NO2 and ozone, but given the literature at the time that we started this project, and even the emerging literature, it's these smaller particles, right, that seem to be pretty harmful. And so we know that prenatally, the routes to having small particles enter the body are different prenatally than postnatally, right? Prenatally, the exposure is through the mother's lungs. It can go into the bloodstream and the placenta, and then pass into the child's or the fetus circulation. Of course, it can also just act on the mother's maternal immune activation, which could have its own profile of effects. Postnatally, there's the direct and indirect routes. The direct route, still is debatable, but I think there's belief that some of the very small particles might translocate directly via the olfactory bulb. And then there's what I call indirect, which is, going into the lungs. And the smaller the particle, the more deeply, right, they go into the lungs. They can then pass into the bloodstream and affect the blood-brain barrier. Some of them can actually pass the blood-brain barrier, it's thought, and influence the brain. Once it gets to the brain, there's been a number, and again, this coming mostly from wonderful animal studies about the mechanisms of neurotoxicity. So we know that exposure is thought to activate microglial cells and astrocytes that may lead to neuroinflammation or changes in myelination. We also know that some air toxicants, again, depending on size and the actual composition, but also influence the blood-brain barrier, making it more leaky. And there's a bunch of work by Deborah Corey-Schlechte's work showing that this also could lead to metal dyshomeostasis depending on what is actually being breathed in. So this was really, a novel neurotoxicant from my mind, maybe not for everyone on this call, but at the time thinking about environment and brain health when we started this work. And so I also really was interested because there had been some really great studies showing that air pollution exposure and was related to delays in psychomotor development, lower IQ, risk for autism, and even anxiety, depression, and attention problems from some of the existing cohorts that had been studied. But I often found the literature focused on either the maternal and the prenatal period, and then they would often talk about the adult response. And at the time, now mind you, now it's much more prevalent, and there's a lot of studies to look at adolescents, which is really exciting. There was kind of a gap in once their postnatal exposure and the child and adolescent periods. And so that's where I got very excited about the potential to study these questions. More and more, of course, there's also the link between air pollution and mental health outcomes specifically. This wonderful review article in 2022 went through the literature to suggest both in animal and human studies that there were 111 mental health studies showing associations between some sort of air pollution and mental health outcomes. And they went on in order to also look at whether or not they could identify certain structures, and found that frontal limbic structure and function was implemented in a number of these studies. But again, concluded that little attention had been given directly to the exposure window of exposure happening in child and adolescents, in this period that I'm very, very passionate about. I will also say there were a great number of studies. We wrote this very small, I don't even know you can call it a systematic review when there's only 6 studies, but a very small systematic review when we first started this work in 2019. And there had been a handful of wonderful studies to prove point of principle that air pollution was harmful, including the wonderful study coming out of Cincinnati, the CCAPS study with the elemental carbon from traffic. But there was enough literature to suggest that these things could be harmful, but questions remained, which was, which brain mechanisms, which postnatal exposure effects might exist that might be unique or different from prenatal. Of course, we're still dealing with questions of sources of exposure and exposure heterogeneity. There's also heterogeneity effects in terms of the population who might be at risk. And of course, there's more or less the question that I'm always interested in is, are these effects long lasting? If you're exposed early on, have you set the course, or because the brain is so malleable that there's some sort of recovery that occurs over time? And this is really really, something that's of interest because a number of the cells that we talked about being impacted by air pollution neurotoxicity, PM 2.5 specifically, these cell types, microglia, astrocytes, and a number of other cells are involved in some of the key processes I mentioned at the beginning that continue to ongo throughout the adolescent period. From neurogenesis and synaptic pruning to myelination, and even the formation and maturation of the blood-brain barrier. And so if these cells at any given time are trying to clean up air pollution, or they're being reactive to some sort, can they then do their job correctly in order to have the various systems of the brain undergo development at the right time and in the right manner? And so this really, this work of timing and this idea that these cells do different things at different times and are malleable for so long really make the question up that we need to start thinking about longitudinal studies. Both the timing of exposure matters, but also the timing of brain assessment, because the brain is ever evolving. And so this is where our work really aimed to fill the gap in the literature. This led to our first study, what I call ABCD AIR, but examining the long-term exposure and brain development through late childhood and early adolescence. It's quasi-longitudinal because there are only 2 time points in this first wave of the grant, but with additional funding, we're hoping now to have the lifespan of both exposure and the 10 years of the ABCD study to answer some really important questions. Today, we'll be focusing on what we've seen so far when the kids started the study at 9 to 10, and then a 2-year follow-up. And so that's the work I'll be discussing. So going back to the LED work and the environmental context, and largely using residential addresses, we look at air pollution via estimates that come from Joel Schwartz at Harvard's models. His models, take the EPA monitors. They use a number of land use terms, chemical transports, meteorology, and satellite measurements. They do some ensemble model integration in order to then predict at a 1-kilometer grid daily estimates of PM 2.5, ozone, and nitrogen dioxide. And ABCD as a whole, the levels on average are much lower than the standards, even the recent change in standards for PM 2.5, and they're much lower for both O3 and NO2. So we're talking about low-level exposure in our research. So the first set of analyses, while we waited for the data to be collected, were cross-sectional. So I did say the point was longitudinal. We'll get there in just a minute. But some of our earlier studies looked at air pollution exposure at the residences when participants enrolled. And then we looked at a number of neuroimaging outcomes and behaviors. And we're using a multi-level framework. In the work I'll be presenting, we're doing, except for the first study, we're doing multi-pollutant models, trying to adjust for NO2 or ozone, or use some sort of mixture analysis or covariation analysis. And all of our models, we control for a number of key confounders. And so everything I'm going to talk about today has been fully adjusted and FDR corrected, and happy to chat about any of the questions about confounders at the end. So the first study that we did, proof of principle, we didn't have NO2 or ozone yet. So this is the only one where we're not adjusting for those other exposures. But we were looking at PM 2.5 exposure levels. And you can see here that at the time the EPA was still 12. So I apologize, I need to move this down to current standards. But there was wide variability by site in exposure, again, allowing for this, this question to be answered at a larger population level than any one given MRI study, which is typically, at one location in the past. So what did we find with this first study? We were looking at brain structure. Here's some examples of some of the brain structure that we can get out of these kind of scans. Things like white matter, which is the myelinated fibers, or gray matter, which is the cortical cell bodies and their dendrites, as well as cortical thickness, which is the distance between the cortical ribbon and the beginning of white matter in the brain. And the take-home of the first study was that we found that annualized PM 2.5, so a year of exposure to PM 2.5 when the kids were 9 to 10, was not associated with overall brain size. But rather, and I was a little surprised by this at the beginning, was that there were hemispheric and region-specific differences. And this was seen in all the measures we looked at: cortical thickness, surface area, and also subcortical volumes. Moreover, we found at this one that total PM2.5 mass was not exposed, it was not related to cognition on the NIH toolbox. Now, when we looked at the patterns of associations, what we saw was that there were both increase and decreases or associations suggesting higher exposure was related to larger cortical thickness or surface area, as well as smaller. And these were in a regional specific manner, which you can see here. Well, as I mentioned, this was a little bit surprising. We later decided that we thought this is because of the fact that these brain regions peak and prune at different rates. So going back to the idea that the brain is ever changing, and that as part of the adolescent process, we build synapses and then we prune them away for efficiency. And so because this was cross-sectional, we really are only getting a snapshot, which means that higher exposure could mean you have a delay in this pruning, or you could have an exaggeration in pruning, and that would give you a different pattern. And so this had led to the hypothesis later on, which we looked at longitudinally, that maybe the rates of change in which cortical thickness pruning occur during adolescence were being impacted. The next cross-sectional study we did was looking at diffusion MRI. So most people look at what's known as diffusion tensor imaging. This is a MRI technique in which you're essentially looking for water movement in the brain. We have a lot of water in the brain, and it is either going to move without boundaries, so it's going to diffuse widely, or it's going to diffuse along boundaries in brain tissue. So this can be things like myelinated axons, or cell bodies, whether that's neurites or glial cells. In diffusion MRI or DTI, you take an average of the whole voxel, a voxel being a pixel of the brain, to say what is the overall diffusion occurring, and is it occurring in one direction. So you'll often hear terms like FA, whether it's restricted or not, or mean diffusivity, which is just how much diffusion is occurring. We wanted to go a step further, and so our work looks at what's known as restrictive spectrum imaging, which is a mouthful, but it goes for RSI for short. And what this is it uses more advanced MRI methodology to create a multi-shell so that you can model intra and extracellular diffusion. What this means is that you can then estimate what might be diffusing in a cell, so intracellularly in a long direction within a given voxel. You can also measure the percentage or the normalization of how much is spherical within a cell. And then what we call is hindered, or HNT here, which is diffusion between boundaries of the brain. And so going from an overall summary score of a voxel to percentages of what could be bound in cells and what type of restriction is occurring in those cells really allows for us to get a better tissue properties. This technique, RSI, has also been validated with histology, which makes us feel a little bit better. And so even though we're far removed from this, the interpretation of actual cells is thought to be that the restricted neurite density is this elongation and possibly due to myelination in a voxel. The spherical, or RNI here, which we'll talk a little bit more about, are the glial cells or cell bodies, if you will. And then the extracellular space here is shown in pink. So using RSI, one of the first studies we showed, and this is adjusting for NO2, is that PM2.5 was associated with increases in this RNI component, or this isotropic pattern of diffusion within white matter tracks. You can see the tracks we found this pattern in across the brain, and you can largely see that this pattern was nonlinear. So we saw the biggest increases in the diffusion within this spherical space at higher levels of PM2.5 concentration. And so this is thought to be interpreted as possibly more glial cells. So in addition to looking at white matter and RNI, with white matter thought to be increase in the quantity or morphology of spherical cells like glia, or more spherical than neurites, you can also look at this within gray matter volume as well. And so we wanted to do this in our next study, in which we looked at PM 2.5, NO2, and ozone in a multivariate technique known as partial least squared correlation. What this does is it allows us to look at exposure to multiple pollutants at once and its covariance with various brain outcomes. So this work was led by Kirthana Sukumaran and Dr. Carlos Cardenas Iñiguez, in which we were able to look at how patterns of exposure related to patterns of brain architecture in gray matter using this RSI technique. And what we found was that when you put these in the model after adjusting for everything, that higher exposure to PM 2.5 specifically was driving associations with higher RNI, again, the signal of the spherical shape of water diffusion within the accumbens, the right thalamus, as well as part of the brainstem. Again, this is congruent with this idea of either more or larger spherical cell shapes in the brain. Just to go back a step here, we hypothesized then that PM 2.5 effects so far seem to be changing microtissue in white matter and gray matter within this isotropic compartment, which again would be possibly related to activated astrocytes or glial cells, or even swelling of cells possibly in white matter or gray matter. Also, this is some interpretation, again, we're not at the cellular level, but is based, of course, on animal models suggesting this happens with exposure. So the next part was then moving on to our second wave of data to say whether or not we could see patterns or impacts of PM 2.5 exposure on some of the changes over time over 2 waves of data. And specifically, we really wanted to focus on 2 hallmarks, white matter microstructure, as well as cortical development, again, going back to this myelination and or pruning effects that we know are really important. And we really were interested in these because they're also transdiagnostic markers for, in the brain imaging world for risk for mental health later on. So here we are looking at exposure. We'll get to in a minute where we are now with the exposure estimates, but for these papers, we actually only had 1 year of annual average PM 2.5 still at the baseline estimate of the study. Most of our studies then restricted our studies to those that hadn't moved since that time. And we're looking at imaging modalities here, structural diffusion, which we both talked about, and we'll also briefly talk about some of the functional findings at the 2-year follow-up. And again, we use the same approach, adjusting for key confounders and FDR correction. So in terms of the longitudinal work, if you remember, we had seen that there was some peaking and pruning hypotheses that came out of PM 2.5 associations with cortical thickness in our earlier work. And Aletha DeJesus, who is now a wonderful grad student at UPenn, was a staff member at the time and took on this ambitious project to look at whether or not the rates of cortical thinning and cortical surface area were different. And what we found was that in the areas in red here, largely in the prefrontal cortex, we saw that there was, in fact, an increase or faster pruning, if you will, for those exposed to higher levels of PM 2.5, including over the 2 waves of data. The only opposite pattern that was seen was actually in the entorhinal cortex, where here the average low exposure are here, and you see that the entorhinal cortex is actually growing. But with higher exposure, you actually see that there is actually almost either a flat line with the confidence intervals or a slight decrease in the growth of the entorhinal cortex's cortical thickness over this follow-up period up to age 14. An interesting other analysis that came out of her work was that she also wanted to look to see if whether or not there was interacting effects with neighborhood disadvantage. We published a number of papers looking at neighborhood disadvantage on its own as a possible environmental contextual factor on brain structure and function. And so the previous work here is showing adjusting for the neighborhood disadvantage. So PM 2.5 independent effects, if you will, at least statistically speaking. But she also found a couple of brain regions and where there were these interactions with neighborhood disadvantage. And these were mostly in the temporal lobes and the occipital lobe. And we found that the slope, if you will, for cortical thickness rates of change were steeper for those in a high neighborhood disadvantage context, suggesting that may be supportive of the double jeopardy hypothesis. We then went on to follow up on some of our RSI diffusion work in white matter tracks, and this was led by Devin Cotter at the time, who I need to change this. She has her PhD. She's a postdoc now in New York. And she looked at the associations between PM 2.5, NO2, and ozone on each of the tracks of interest. And I know there's a lot going on, so let me walk you through it. Here's each of the tracks across the brain. And what she has plotted in these heat maps are the effective PM 2.5, NO2, and ozone at age 9. And these columns are the ones in which we see the largest effects. So at age 9, we see RSI and RND effects, these differences in the diffusion properties of the white matter microtissue when they start the study. She also has columns here of the interaction terms. So age by PM 2.5, so is the rate of change different, whether you're exposed to high or low PM 2.5, NO2, or ozone? And you see that these columns are largely blank. And this pattern is true for both male and female. They tend to have the PM 2.5 effects present by age 9 with more of a persistent effect, no change in slope over time. But she also wanted to highlight that the patterns of male and female youth showed some differences. So for example, she found stronger associations with ozone in some of the regions like the forceps minor, part of the corpus callosum, and the cingulate, as well as the cortical spinal tract, where she saw more robust phenotypes with nitrogen dioxide in females, which are shown here in all of these tracks, and only ozone with the central or the cortical spinal tract. And so some differentiation between male and female youth. And this was the first time we had looked in the other studies for sex differences. There's obviously a body of literature to suggest the effects could be different by sex, but this was really the first time in which we started to see this show up in white matter microstructure. Dr. Cotter also went on to study resting state functional connections. So this is a technique in MRI where you essentially are looking at the blood oxygen level signal or brain activity signal in one region of the brain versus another. And you can see how well they are correlated. Higher correlations are thought to be functionally connected regions of the brain, and it can tell us something about intrinsic organization of brain activity. And so she was interested in looking at these patterns within 3 networks specifically: the default mode network, the salience network, and the central executive network. Together, these make up what is known as the triple network hypothesis, which are really important for integrating and segregating from one another when you do mental tasks, and thought to be important for risk for psychopathology. As a whole, we know with age, although most of the work is cross-sectional still, that networks become more integrated within. So for example, areas within any given network start to talk to each other more by having stronger correlations. And other areas of the brain, so other networks, actually become more segregated. So the default mode network become more like itself, those regions, whereas they would do a different pattern as compared to the salience network. And so this is both known as within network integration and between network segregation in terms of how the brain is thought to mature in these resting state networks. So Dr. Cotter then looked at these in ABCD to look at how PM 2.5, NO2, and ozone associate with changes over time in these networks. And first, because there hasn't been a lot of longitudinal studies, she first plotted what is happening with age. She found the default mode network and the frontal parietal networks, also known as the central executive, were kind of integrating with each other with age as expected. The saliency network, however, was actually becoming weaker with age, as well as integrating more with the frontal parietal network. And so a little bit different than the cross-sectional work. In terms of what exposure was doing, she found that higher PM 2.5 exposure during 9 to 10 years of age was related with less between network segregation with age. So you want these networks to segregate. You want them to become more specialized and disentangle from one another. And she found that there was less of this to be occurring with higher exposure, especially for the default mode network and how it interacted with both the salience network and the frontal parietal network. For higher NO2 exposure, she saw that there was more between network segregation with age. And with O3, she found that the within default mode network integration over time was affected. So these studies have shown that at least quasi-longitudinal, that air pollution, at least at this transition period of early adolescence, really might influence both cross-sectional patterns of brain maturation, but also the rate in which some of these processes change over time. Although, some of them are stable, like the white matter. So there seems to be some brain phenotype specificity and what's affected and how it's affected, which might go back again to this idea that these processes are malleable for different rates of time and in different brain regions in order to achieve, cognitive and emotional regulation processes by adulthood. So that was some of the work we did with the overall PM 2.5 total mass. But the last couple of years, we've really been working to try to move beyond total PM 2.5 mass. As many of you will appreciate, right, PM 2.5 is a heterogeneous mixture that varies by geography, by season, by emission sources. And so, of course, we're worried that by just looking at PM 2.5 total, we might be missing the complexity of this. And also, knowing the sources might actually also help us regulate certain policies for better brain health for all. And so this work has really led us to working with Dr. Rima Harber here at USC, who is an environmental scientist, among many other things. She also runs the Climate Center here as well. She worked with one of my staff members, Kirthana Sukumaran, in order to start to look at whether or not we could look at PM components and sources. So similar to Joel Schwartz models of total PM 2.5, NO2, and ozone, he's developed 15 PM component models that we then were able to link to the ABCD residential addresses when the kids were 9 to 10. These ones are also at the resolution of 50 meters in urban areas, and they also are at the resolution of monthly, so not daily. But we nonetheless averaged them across the annual year to look at an annual estimate of 5 carbon ions and 10 trace elements. And so we can look at each of these components as their own or a mixture effects. In addition, we worked with Rima to try to see if whether or not we could kind of estimate what sources these might be coming from. And so she helped us perform a positive matrix factorization in which we were able to really come up with 6 known sources from the literature across the 21 sites using the ABCD study. And so this included things like crustal material, which was driven by silicon and calcium, ammonium sulfates, driven by ammonium and sulfate, biomass burning, traffic, ammonium nitrates, and then this industrial/residual fuel and oil burning. And so these 6 sources that Rima and Joel Schwartz had helped us put together could then be used in our models to say whether or not various mixtures or associations with the components and or the sources map onto differences in brain structure, but also in cognition. So the first study that also did the positive matrix factorization also looked at whether or not there was an influence on cognition using the NIH toolbox. And so in order to do this, we used the mixture modeling approach, weighted quantile sum regression, in which we looked at whether or not the 15 components influenced general cognitive ability, learning and memory, and executive functioning based on composite scores from the NIH Toolbox Cognitive Battery. And we found that in every case, there was a negative association. So higher these kind of mixture effects of PM 2.5 components, and the poorer these scores were. But what was interesting was then looking at the actual weights of each of the components, we saw different profiles for different cognitive patterns. And the most robust one, and so the one that I would focus on the most, given that everything we ran, it showed a really significant association, was that higher ammonium nitrates was linked with worse learning and memory performance. We then, of course, interested in the components and the sources, wanted to move on to see, well, learning and memory is involved or driven by a lot of hippocampal processes. And so I had a postdoctoral fellow who has joined the lab, Dr. Rosario, who's been looking at then whether or not he can disentangle the PM 2.5 components and the sources and how that relates to hippocampal structure and function. And so his work has really used going back to the partial least squared correlation analysis, looking at the 15 components or the 6 sources. And what he's found is that some of the components, copper and zinc, for example, related to smaller head and body and tail proportions in the hippocampus and ABCD study participants. And in terms of RSI, you also looked at RSI in the hippocampus, saw that patterns of exposure were linked with overall decreases in the extracellular space, as well as increases in free water, possibly reflecting less coherent structures. And then, of course, he replicated this with the source loadings, showing that traffic and industrial sources were accounting for some of the head, body, tail findings, and the ammonium nitrates and biomass burning were accounting for some of the RSI patterns that he was seeing with the profiles of the components. The last body of work I want to talk about was the first study to try to combine the thinking about sources of exposure and the longitudinal patterns that we're seeing. And specifically, this work was done by Dr. Katie Bottenhorn, who's now a professor at FIU. She was interested in studying whether or not the sources of PM2.5 exposure impact cortical microarchitecture development. So we talked about longitudinal white matter patterns, and we also talked about using RSI and gray matter. Catherine really wanted to look at whether or not PM2.5 influenced RSI and gray matter across the cortex, something that might be a little bit more closely related to cortical neurodensity and the pruning that goes on during this time. So she had done a great body of work to just show in ABCD as a whole what the patterns of RND and RNI, these microstructural properties, are doing over time in ABCD. And she found that cortical neurite density, this RND pattern, was largely decreasing during the developmental period from 9 to 14 in which we were following the kids. And so she wanted to know if sources of exposure influenced that pattern. And what she found, again, using a partial least squared correlation analysis, is when she put in the sources of exposure, and she also did this with components, that really there was a widespread association in the increases to exposure to specifically sources of biomass burning and decreases in this cortical neurite density and the rate of these decreases over the 2-year period. And so suggesting that there is possibly a disruption to the neurite density patterns in these kids with higher biomass exposure. So overall, this work has shown together that as a nationwide study, that low levels of PM2.5 and some of its components can influence brain structure and function across the US. There seems to be regional and network specificity, and this, I believe, is really stemming from this dynamic neurodevelopmental trajectories we know already exist and that vary by brain region and the pace of which develops by system. Of course, other than the mixture effects with cognition, we really aren't seeing as much effects with behavior as I would have predicted, or I hypothesized even in my grants. And so part of me is wondering if whether or not some of these smaller brain differences might act as early biomarkers for future problems, or maybe they're just epiphenomena. But one of the reasons why they might be biomarkers for earlier problems is that we did originally look at whether or not PM2.5 and NO2 related to emotional problems from 9 to 13 years of over 3 waves of data in ABCD, and we didn't see an association. At the 4-wave data point, we had a colleague in ABCD show that not the annual average, but the number of days above the EPA standards at the time, which was the 12 micrograms per meter cubed, was related to increases in internalizing symptom trajectories, possibly suggesting that it might not be the overall exposure, but kind of these deviations, or if you will. The other reason why I think that there still might be an early marker for later problems comes from some wonderful work by the UK longitudinal twin cohort, where they looked at pollution exposure and found that it was not associated with mental health problems at age 12. So exposure at 12 was not related to health problems at 12. But the exposure at age 12 was related to increases in risk for depression at age 18, as well as the risk for conduct disorder later on. And so it's possible, right, that with enough exposure, and we're only looking at a snapshot of exposure still, but as we build upon this to look at cumulative and sensitive periods, it's very possible that some of these problems might emerge later. Of course, we haven't even really gotten through the hump of the age 14 where most problems start to occur. And so even more prevalence of these problems will occur over the last couple of years that we've been studying in ABCD. It might really be when we might see this elevated odds if it does exist. So with this, I just want to kind of summarize that our work is only a small body of work that's come out the last 20 some years. And so we wrote an updated review paper in order to capture some of the studies that have been done and what we have found. And so I think there's still so much work to be done in terms of both context and of exposure, but also timing and the interactions with other contextual factors. And so in addition to that, I think that our work is really looking forward to sensitive periods and cumulative versus acute exposure patterns. We're really interested in outcome-specific temporal relationships. For example, you might not expect that exposure during early childhood would have the exact same fingerprint, if you will, on brain development as later on. And so this work hopefully will continue to be funded by NIHS